For today's activity, calm down, pass around. You can get the tracing paper as well. Not tracing paper, the graph paper. Everybody got it? Yes. Got it? Okay. Okay, so for today, we are going to be doing, kenapa? Tak ada lagi. Jangan sorok, give to your friend. Okay, all good? Breathe. Nak marathon, nak sprint ni. Bukan marathon, nak sprint. Right. So today we're going to be doing two activities. The first one is the leaf area determination, which you are going to be utilizing two methods. The first one using the leaf area meter, which you have dealt before a few weeks back. And the second one is using the graph paper. That's why you are provided with graph paper. So the idea is to compare between the two methods, the manual method versus the automated method. Are they going to be yielding the same results, or are there going to be some kind of discrepancy between the two? So that's what you're going to find out and make comparison and discuss a bit more about it. Maybe one method is more suitable for other in certain cases or events, right? And once you're done with that, very quickly move to the second activity, which is, this is actually in your lab. I should mention, right, this is lab number what? So for the leaf area, that should be week in your manual. It's labeled with week 3 and week 4. Yep, week 3 and week 4. That's for the leaf area determination activity, right? For the second activity, the growth analysis, which is labeled as week 2 in your lab manual, we are going to be doing that afterwards. So this growth analysis, actually it requires several weeks because you need to wait for your seeds to germinate and grow before you can collect the organs for 2 weeks. Remember your growth analysis formula? There's T1 and T2. So that T2 is the second week. T1 is the first week after your plants have grown. So I expect the experiment going to take time about maybe 3 weeks. Yeah, so this week you grow, and next week we see whether it's big enough or not. Because this is corn. Corn is quite fast. If it's big enough, you can harvest some of the biomass as your T1, right? And then the following week, harvest some more for your T2. Then you're done with it, okay? So for this activity, simply involve germination only. You got your seeds, you got your germination, okay? I'm not going to explain just yet what's to be doing here. Let's come back to the first leaf area activity. So, I just want to save time. Do you still remember how to use that machine?
Yeah.
It's quite easy, right?
Yeah.
Right, so it's rather easy. The specimen that we're going to be using today is this plant. What plant is this? Tapioca. Really? So the common and the vernacular name for it is ubi bunga. Yeah. So maybe it's the same tapioca esculenta that you know. Maybe the esculenta is very variegatum because it's got variegation on it, right? So use that machine for your first determination of leaf area. Being a science student, actually, if you use this whole thing, it might not be able to capture the entirety of this area because this leaf came from the fridge, you know, over the holiday. So they kind of a bit bending this way. Yeah. So if you get specimen which may be a bit not flat, maybe something like this, not flat, and you want to save time, you can actually remove it, the finger. Remove the finger, but you need to remember how many finger that you have removed, and then you can just add up the leaf area of it. Okay. Can you turn on? Somebody turn on that thing. Run it through. Is it running? Can you run this? Run this. Salah. Patutnya I do not detach first. I just want to see whether it can capture. Can it? Okay, it can capture. Right. You know what? I got a better idea. Just to diversify your result. Before you detach, you get the whole of this first. Okay? Just insert the whole of this first. I hope it can go through. It can. Yep. And then detach. Right. So that's the first method to do. The second method would be to quantify it manually using the graph paper. Do you have your graph paper? Okay. So to do that, so you can see that the graph paper is actually got these small grids. The smallest grid. Do you bring your pencil?
Yes.
Okay. Where's my pencil? I thought my pencil was. Okay. Let me think. You know what? Even though I explained that first, I think in sequence you should be doing manual first, because this is non-destructive. Okay. However, this leaf is rather wet now. Use the paper towel. If you need more paper towel, it's over here. Use the paper towel. Dab dry the leaf. If it's cold, it's fine. Not wet. Okay. Because if your leaf is rather wet, you are not going to get a good result when determining this thing. Is it okay? Still wet. Okay. Okay. This is right. So how you want to transfer this to here? Very easy. Just use the tracing method. You can either use the top side of the leaf. I need to explain the top side of the leaf. So the leaf got two sides of it. So you got the upside of the leaf. We call it a daxial. And then we got the downside. We call it a baxial. Usually, most leaf, they have protruding veins on the abaxial. Okay, mostly. But in the case of this leaf, you are free to either use either side, a daxial side or a baxial side. So the way you do it is, you take any side that you want. Let's say that I want to use the abaxial side. Put your dry leaf on the flat surface. Put your tracing paper on it. Oops. Yeah. And do not move. And then start tracing. And while doing this, you might want to shy away your friend. Okay, so can you see? So what you are going to quantify is the outline of the leaf later on. If you can manage the whole of this leaf, the palmate shape of this leaf, to this one graphing paper, that's fine. That's good. Okay, so do that. If you can't, you can remove the finger one by one. It's up to you. So you might want to use a small one, medium one. Yeah, yeah. Okay. So once you're done with this, then you can proceed to that equipment. Run through this entire palmate leaf, and then you can remove one by one and see whether there's a difference or not. All right. Okay. So to quantify this, based on the grid, so you have the small grid. 1, 2, 3, 4, 5. So each of these, the small grid, that's equivalent to 2 millimeter. This one equals to 1 centimeter. This whole the big square, that's 2 centimeter. So if your leaf kind of occupy that region, and when you zoom out, let's say that we are looking at this thing, one small section, and your leaf is kind of like that. The ruling is if it's less than half, less than half, it's equal 0. If it's more than half, equals to 1. If it's equal 1, equals 1 lah. Okay, so you can, the situation, you can be something like this, something like this, or something like this. Okay, so is this 0, 1, 1? 0. This 1, 1. Easy. So now you get your, that's 2 mm. 2 mm. Oh, no, not this one. This is 0. This is 2 mm times 2 mm. So do you just occupy your 4 square millimeter? That's the smallest square in here. So 3 types of square: smallest square, medium square, and the big square. All right. Okay. So I advise you to do this quickly. You don't have to quantify today. You can quantify when you go back to your place. Okay. Then you proceed with that. Is that clear? Is that clear? Can you do it now? Yeah. All right. Okay. So you can move that. So let's finish that in about 40 minutes. Then we move to the second activity. Okay. Right. I am here. You can ask anything that you see here. This is for the smallest square. You got 3 levels of square, right? Not for the medium, not for the large square. This is for the smallest square in the graphing paper. So to ease your visualization, maybe you can use multiple pen color. So for the smallest, whenever it has reached either this or this, just simply tick with maybe red color. And later on, you can quantify easier because just count the number of tick. Okay, so you can use your pen color or something to assist you. I know it could be harder to look at when you use the carbon pencil, but you can use the pen to tick the square that has achieved either this or this. Tick first, count later. Okay. Okay, Kami. Kalau ada aku suruh dia orang buat kat luar je. Ada lubang? Ini pun ada lubang lah. Okay. Okay, 8:46. Try to finish within 15 minutes, okay? Then we'll move to the second activity. I expect you to do a decent lab report, okay? This is not for me, this is for you. The more you are becoming expressive with your lab report, that's for your own good. Or you can ask me afterward because I need to show you how to do the. Okay. Maybe you should come a bit closer to me. Throw away your computer, Tracy. Throw away. Throw away. Throw away. Throw away. Throw away. Throw away. I need water sauce sebenarnya. Kau tolong bawa ni ke sinki. Nak pergi dekat dengan sinki. You can come here. Sini. Sinki kat sini. Bangunlah sekejap.
Sadar pergi tadi.
Spread around. You can spread all the way sampai sana. Nanti pergilah balik kat tempat. Duduk.
Biar nampak korang air. Kalau di sana kan nampak korang air.
Cepat. Move quickly. Okay, so for this experiment, we are going to germinate. What seed is this? Corn. Kacang panjang.
Corn.
Jagung.
What's the Latin name for corn? Zea. Z-maize. Maize you can understand because it's maize. What about the Z? It's referring to what? Yeah. It's referring to the pigment in corn, zeaxanthin. Why do you think corn is yellow in color? Carrot, orange because beta carotene. Corn is yellow because zeaxanthin. Right? So they turn the name into Z-maize. Right. Okay. So to do this, you can see that the media is rather dry. Abilah macam mana lah kau nak ni? Do not put the seeds straight away. You need to wetten this first. So how do you do it? I advise you to create a hole first, a well. Lepas tu
ni Mat, tak puasa boleh macam.
Kenapa? So maybe you can ask your friend to help. Turn this box while you have this water running. And then give it a good bang. Do it again. Usually if you do it 2 rounds, the moisture should be enough. Why? Why do it this way? Oops, sorry.
Evenly spread.
Yeah, because this is sand. Sand is super porous. You want the water to go all the way to the bottom. Nampak? Kalau tak buat properly, sebenarnya kat bawah tadi yang kering lagi. Tapi sebenarnya dah kering. Ya, ada lagi kan? See?
Sikit.
Yep. So until what consistency should you add your water? Sampai dia boleh jadi ball. More or less. Like this. Okay. Not too much. I don't want to see it jadi aquarium benda ni. Okay. Not aquarium. Okay. Right. Okay. Let's do this. Any group nak buat? Let's see. Group mana nak buat? Buatlah. Kan I have shown. No need to break it. Let's put the water. Sometimes when you put the water, dia akan break. Slow, slow. Maybe one of you hold this, and then your friend can each so that you can turn. Yep. Turn. Mesti tak pernah bakar biskut kan? Okay, that's one round. Sana, jangan kat sini. This is a weak point. Bang, bang. Either end.
There was a little like.
Lagi. Pusing lagi. Okay. Break dapat macam tu. Nampak? Ada lagi tu. I think you need to add a bit more moisture. Maybe do it half round. So this is why I want you to see this. Sometimes 2 turn is enough. Sometimes you need a bit more. So maybe for this one you need half, a bit more. Weh, jangan mana?
Yang lepas awak sebut kat saya dia.
Kenapa kau bergaul macam tu? Tadi I didn't do that pun. Kan dia elok je. So, kali buat macam ni.
Kamu je
Cik, kering. Kau tambah lagi. Tapi yang banyak sangat. Yeah.
Kecil.
Okay, so you can see that after the consistency has been achieved, you can form a ball like this and the color is getting darker. Okay, right. So let's say that's done. Let's see how do you do the germination. So you are going to do the biomass harvesting for 2 weeks. So it's a good idea now to do this into 2. Maybe you use a paper or something. Can somebody get me a graph paper? The paper. Remember, okay, this thing. Why is it pink?
Ada hormon.
Ada hormon? Ada?
Fungicide.
Apa fungicide dia? Apa fungicide dia? Yelah. Corn, what color is corn?
Yellow.
Is that yellow?
No.
Okay. Why is it not yellow?
Because it's coated with fungicide.
Fungicide. What's the name of the fungicide? Tiram. Thiram. Tiram. Have you taken your amalan ladang? You should learn during amalan ladang. Kita tak ambil?
Maladang December.
Yeah, but we only learn until climbers. Ada.
Until?
About plants.
Until?
Plants only. No plants.
Oh.
April.
So, tak belajar habis?
Tak masuk lagi.
So, divide lah 2. This is just an example, okay? For however you want to get creative. So, for week 1 and week 2. Since this is treated with tiram, make sure that you wash your hand with soap thoroughly. Otherwise, nanti engkau yang jadi apa?
Mayat.
You are the one going to turn pink.
Kan itu rendam ya.
Actually, that's what I want to show. Ada cling wrap. Maybe. Can you get a cling wrap? Mi, ada cling wrap. Plastic wrap. Ambil sat. So the reason kita tak nak rendam ni overnight because we don't want to remove the coating. Because while the seeds are germinating, the fungicide is still protecting it from the fungus because it got all spores in the air now. Okay, so if you soak it this way, can you see the color now?
Yes.
You are actually removing the fungicide, the protection for the seed. So what do? Adalah we can trap the moisture in the germination box. Okay, can you remove it? Nanti basuh tangan. Remove the moisture. Air.
Buang je air.
Tak banyak lah.
Habislah tu tapi.
Ada Mi? Ambil tu dengan gunting.
Doktor. Kena jatuh sini.
Angkat lah. Aku cakap aku tak nak buat biskut, kau tak percaya. Apalah, ni ada kau nak buang air. Apalah, ni buat macam ni je.
Saya buat benda sama dah tadi.
Yelah, tapi kenapa aku habis? Payah sangat.
Takut.
Takut apa?
Sakit jatuh.
Rendam je.
Okay. So how many seeds? I wonder how many seeds. Cuba kira. Okay, can you come? Put it half. Apa? This is another reason kenapa kita tak soak ni. 31. So 15.
15, 16.
Yeah. So you can use your judgment, you can use the ruler. What? You can do the same for this as well. Pergi bagus tu. The other group while doing it, ambil lah. Easy. Boleh guna singgih sini.
Can it be 15, 16 doktor?
Boleh. Snap some images so that you have material for your lab report. The other group you can use that sink. The sink got the hose. Ni. Sorry. Ni. Tepi ni. Use this. Just give a good rinse sekali saja. Don't.
Siapa ambil bekas ini tadi?
Tak keluar air. Friends dah letak seeds dalam ni. Okay, you add a bit more air. Jangan tak atas tu. Letak dekat corner. About that much. Let it down. Yes, because kita tak nak kacau seeds tu. The seeds need to stay in place. About that much. Tutup. Okay. So give it a good swirl. Left, right, front, back. Then we use the cling wrap. Pull a bit more. Yep. Yeah. So we do it 2 times because the cling wrap is not white. So do it this way, do it this way. Are you okay? Okay. Do one way.
I don't have the label.
Oh, label. Sorry. That's not label. So you need to put the label for both section, for T1 and T2, which is week 1 and week 2.
Boleh guna ke?
Oh, kenapa banjir?
Tak tahu rotan ni. It's dry down here. I already mix it thoroughly.
Okay lah, tak apalah.
Maybe bila saya bawa sini dia jadi macam tau. Dia jadi wet.
I think that group macam send it terlebih. Can you donate one hand ke sini?
Bagi.
Sikit je. Ambil lagi satu kat sini. Dah. Ambil. Okay, then use the pencil to poke some holes. Maximum 5. Don't. You can use the pencil to poke.
Ada warna sampai takut ni macam
Okay lah ni. Tak apa, nanti dia germinate, you going to remove this anyway.
maximum 5.
Yep, maximum 5, 6. Jangan banyak sangat. Nanti moisture loss. Okay. Jauh-jauh sikit lah, jangan dekat.
Not enough.
Bang, cepat. Orang dah nak siap dah. Cepat.
Oh ya.
Okay, wrap. Tarik lebih sikit sebab nanti you need to wrap on both sides. Yes. Tak apa. Yang tepi is if not occupying, you use the other one to go this way.
Ada.
Okay, mana Amy? Tanya kat dia letak mana?
Dia dah turun dah.
I think nanti Mr. Helmi can place dekat luar. Before you leave, another trick to make this grow faster, leave some moisture on it. Take some water, splash on it. On this. So it becomes cooler.
Ya. Kita tengah kan.
So this means kalau dia cooler, dia akan jadi cepat lagi cepat.
Tapi if it's not treated dengan fungicide, nanti dia fungus. Since this got fungicide, you don't have to worry about that.
Apa dia dah ni tidak ada.
I think that your sign macam nampak kering sikit lah. I think it doesn't hurt if you put a bit more air. Dekat maybe.
Jago.
Di lubang tadi.
Jangan besar, buka lah tepi. Sebab tu kita guna clean wrap. Kalau kepala buka, So add water, lepas tu gerakkan. Dah. Cucuk 5 6. Jangan dekat-dekat.
Okay, dah. Sebelah apa sebelah? Dah apa sebelah?
Okay. This group is done. You do that. How's your soil? Does it look okay? Hmm. So this is one side. Lepas tu buka this side. Okay. That looks good. Sekejap ya. Put your group name as well. Mesti kau tak letak tarikh kan? Letaklah tarikh. Kan hari ni hari germination. Letak tarikh. Dah. Okay. Apa lagi grup ni? Orang biskut dah nak siap dah. Kau baru nak buat lubang.
Korang tengok resepi yang TikTok doktor.
Kita ikut resepi.
Episod aku tunjuk demo dah habis dah. Tepi mana? Tunjuk kat dia orang lah. Nanti nak letak mana. Group that is done. You can go to Mr. Ami. He will show kat mana nak letak mana ni. You can splash some water tau atas ni. Yeah. Find a water hose. Speaker tu. Janganlah guna air. Aku ambil air kat luar. Kat luar ada air. Mi, tunjuk kat dia orang, Mi. Ni group ni. I'll go out sekejap. If you're done, you can follow. Aku sini. Meow. Say hi to camera. Meow.
Tari ada, nama ada. Tapi saya berpegang pada saya belum lepas.
So, ambillah air sikit dekat Encik Aizan so that you can splash on this. Air tu. Kan ada 2. Oh, panjang. Apa jadi lagi 2 grup? Pelan-pelan. Yep. Itu je. Grup ni elok je. Have you poked some hole? Yeah. Okay. Meow.
Boleh tukar wrapping doktor?
Boleh tukar wrapping? Kenapa? What's wrong with wrapping? Nak tukar pergi ambil wrapping. Ambil wrapping baru. Okay, ask your friend bawa wrapping sini. Tak payah bawa ke sana. Bawa wrapping ke here. Ni buat macam ni, nampak? Kemas dia buat.
Wrapping sikit lagi. Tadi kan.
Tu korang kena demo kat dia. Mana lagi 2 grup? Ni tolong panggil yang lagi 2 grup tu sini, budak ni. What time? Okay, sikit. Kata habis 6:45, right? Stick to the plan. Tapi of course lah, dia kena aku masuk campur. Okay. Mana lagi 1 grup? You should be 4, right? So why do we do it this way? Aku cakap kat sini, lepas tu mic kat mana? Lepas tu apa terjerit-jerit kat dalam. Group apa yang struggling sangat tu?
Buat kat sini. Bukan kat air. Oh.
Nanti ada bekas bawah.
Dia orang buat bekas tapi dia binatang. Tapi Aisyah dia cepat.
Cepat. Nanti ada kelas kan pukul 10 kita 9:45 kita nak habis dah.
Masukkan kat dalam juga.
Dah.
Kita usah skop. Lightning kau tu.
Main tu main. Tak macam ni. Tak ada je.
You know what? Why don't you remove ni ke sini sikit sebab shading nanti tak dapat cahaya. Remove? Remove sini. Sebab ni ada pokok kan? Nanti dia shading. Move it. Let's press it. Over here. That group tak nak move ke? Sini. Situ ada shading. Nanti your class not doing well.
Tolonglah group tu, tolong group tu.
Group tu tak ada siapa kisah.
Tolong dia, bawa sini.
Okay. Alright. Okay. So before you go back, let's I'll just explain a bit about this way of germination. Remember, there are many types of germination media. You can use soil mix. That's not soil. We call it soilless culture. And sand is one of the soil types. Have you taken your soil science?
Yeah.
So how many types?
Many types.
Remember you learned the soil triangle? That triangle based on clay, loam, sand. So the combination of this will give you the type of the soil. So this is sand je lah. Sand. The thing with sand is, number one is super porous. Super porous, that's good for germination because the seeds radical, when it's emerging, it's not going to be struggling a lot. Grow very readily. However, this soil or this media, since it is 100% sand, it is almost inert. You know inert? It is without nutrient. And you're going to leave this for 2 or 3 weeks time. So that's why on Wednesday, we're going to be putting some fertilizer to it, which also coincide with your lesson, small lesson, about nutrition on Monday, on Wednesday, right? So be on time on Wednesday. We'll learn a bit about plant nutrition, and then you use your fertilizer to fertilize this. Okay. So why do we wrap?
To hold the moisture.
To hold the moisture. To do what? To achieve what?
Humidity.
Humidity. What about it? Humidity saja. Remember you learned about RH? Have you?
Related humidity.
Relative humidity. What's that? Don't forget itu masuk exam. So relative humidity is the amount of moisture that is being held at the current temperature, at the current sea level elevation for that space. Meaning that if the space is able to withhold maybe 100 liter of water, and then the equipment measures only 80 liters of water that is present, it shows your relative humidity is only 80%, 20% to go. Okay, so we increase the humidity to up to 100% here to facilitate germination. Okay, because it is common for our tropical area, if you expose the seeds without high humidity, that is going to cause seed desiccation, the drying of the seed surface. When that happens, the embryo can actually die, and we don't want that to happen. So we trap the moisture to increase the humidity so that the entirety of the seed, let's say that this is you got your seed, the surface of your seed is in contact with 100% of water. Okay, but you still need oxygen. That's why you cannot soak your seed. You let it in contact with 100% water vapor. So that will enhance the seed germination, and also to ensure that there whenever the seeds is requiring nutrients, the moisture can dissolve some of the minerals in the sand. It's not a lot of nutrient, just enough for the seed, because at the first week the seed is not photosynthesizing. It's actually using the what's the food reserve in this corn called? Is it endospermous or non-endospermous seed? I suggest you go back and revise that lesson, because you're about to do the lab report, right? You need to do the discussion. Yeah. Okay, right. So on Wednesday, let's see. If you find out some of the seeds are not germinating, we can quickly do the re-sowing. I hope everything is going to be just fine, so you don't have to do that. Right? Okay? All right. Is that clear?
Yes.
Is that clear? Are you done with the leaf area?
Yes.
All right. Okay. So I'll see you again on Wednesday.
Alright.
Right. Okay. And pray for the best.
So doctor, by
Yeah.
Wednesday we open the ticket, right?
Yes. We actually you can lift a bit more. Yeah. Because this is corn. Corn is growing super fast because it's C4. In nanti kat photosynthesis you will learn different modes of photosynthesis. Photosynthesis in corn is special. It's super fast. Yeah. All right. Okay. All right. So we'll see more of this again on Wednesday. Right. Yeah. All right. Okay. See you. Don't traumatize the cat.